Thank you for joining us today. My name is Randy Dyer, and I'm the Senior Director of Product Marketing for Purigen Biosystems. Before we move on to our guest speaker, Dr. Sankar Mohan of Canopy Biosciences, I want to tell you a little bit more about our company, our technology, our products, and most importantly, our new FFPE Complete Purification Kit that enables you to simultaneously extract both DNA and RNA, including microRNA, from the same FFPE sample, using one simple automated workflow. Purigen Biosystems was founded in 2012. Our mission is to develop products using our innovative technology to help you get the most information that you can from every sample, especially those samples that are rare or limited, the samples that you can't afford to waste. Our technology was developed by a team at Stanford under the leadership of Professor Juan Santiago. We're headquartered in the Bay Area of California, and we are backed by investors that include 5AM Ventures, Agilent Technologies, Roche Venture Fund, and Coda Capital. Our Ionic system uses a technology based on isotachophoresis. Unlike most other nucleic acid purification technologies, separation in ITP is based on charge, or specifically the electrophoretic mobility of molecules in solution. This doesn't require any use of a physical substrate to bind the nucleic acid for separation. There's no need to wash away impurities or to then elute your nucleic acid off of a physical substrate. Instead, the separation happens freely in solution. So you don't have to worry about the nucleic acid not binding to the column or bead, whatever physical substrate you're using. You don't have to worry about it getting washed away when you then try to wash away impurities or not coming off of the substrate when you need to. The result from using isotachophoresis is higher yields of more intact nucleic acid, and all that's coming from a much easier workflow. This diagram is a simplified representation of what happens to each sample during isotachophoresis. Following lysis, samples are loaded into wells on a fluidic chip. Those wells are connected to fluidic channels. The channels contain buffers that are designed to isolate and separate nucleic acid from other molecules or impurities in your sample. Once current is applied, all of the negatively charged ions, including nucleic acid, start moving down toward the positive end of the channel. As that movement happens, the nucleic acid can't move past the leading ions or the faster moving ions in our separation buffer. It can also not move slower into the trailing ions or the slower moving ions of our cathodic buffer. So effectively, nucleic acid becomes sandwiched by these two buffers as it moves down toward the extraction well. Also, the nucleic acid is being concentrated and gathered into that zone as it moves to the extraction well. So that enables us to collect all of the nucleic acid in the sample and deposit it into the extraction well for you to collect with a pipette tip. What's nice about isotachophoresis too is that we can control that extraction buffer. So we can ensure that your nucleic acid is deposited into a buffer that's going to be compatible with your downstream assays. Here's an animation to show isotachophoresis as it happens on the Ionic system. Here you can see the chip on the Ionic system. I'm going to pause it just to kind of give you or review the layout of the chip. On the left-hand side of the chip are a series of wells that are used to fill the channels below with buffer. On the right-hand side are similar wells that are used to load sample into the same channels. Here we've used a dye to show sample or lysate that has been loaded into each well. Normally we don't use dye, but for demonstration purposes, we do. Once the run starts, you'll see that that smear of sample is going to start to gather. And that is that concentration effect that I spoke about earlier. And you can see that as the run continues, that band or nucleic acid gets concentrated into a tight band before it enters into the well where you can extract it. This cartoon or animation shows this in a little bit more detail. The blue ions are the faster moving ions, and the red are the trailing ions. And as you can see, the impurities are moving away from the nucleic acid into those trailing ions. And the nucleic acid will continue and concentrate as it moves down the channel into the extraction well, where you can use a standard pipette tip to then extract it. And the result, as I said, it's more intact, higher yield, high purity nucleic acid. Now I want to tell you more about the Ionic system. There are two major components to our solution. That is application-specific kits and the Ionic instrument itself. Each kit contains reagents and fluidic chips to process 48 samples. And that's 8 samples per run and 8 samples per chip. The Ionic system itself is designed to be on the benchtop. It's about the width of a laptop, the depth of 2 laptops, and the height of a laptop and a half. You can operate the system from the touchscreen, so there's no need to connect a separate computer. And as I'll show you in this presentation, there are several advantages, including a simpler workflow that produces nucleic acid in a higher yield with higher quality. To use the Ionic system, you place a chip on the instrument. Use a multi-channel pipette to load buffers from the kit onto that chip. Once you do that, you press a button on the touchscreen to start a priming cycle. It takes a few minutes. After that, you'll load lysate from 8 samples onto the chip. Once the samples are loaded, you start a purification run. After about 50 to 60 minutes, you come back and you can extract pure nucleic acid from the extraction wells using a standard pipette tip. The chip requires 200 microliters of each sample for a run, and the extract is produced in a 50 microliter volume. The time to lyse the samples is going to vary from kit to kit, but the purification time on the Ionic system will typically be around 60 minutes. But the most important thing is the hands-on time is quite minimal. You're really only using a few minutes to load buffers, start the priming, and then load samples, and then coming back and extracting the material. For FFPE samples, our first 2 kits were designed to produce high yields of high-quality RNA or DNA. The FFPE to pure DNA kit was launched first, and that was followed by our FFPE to pure RNA kit. I want to point out that for the FFPE to pure RNA kit, the RNA that's produced is total RNA. It includes mRNA as well as small non-coding RNAs like miRNA. Our newest kit, the FFPE Complete Kit, combines the advantages of those earlier kits into a single workflow for simultaneous extraction of DNA and RNA from a single FFPE sample. Before I get into the details of the FFPE Complete Purification Kit, I want to talk to you more about our FFPE to pure DNA and FFPE to pure RNA kits, because the Complete Kit really evolved from the work that was put into these 2 kits. Let's start with the FFPE to pure DNA kit first. At the top of the screen, you'll see the steps that are typically involved with isolation of DNA using a column-based kit. For column kits, you typically start by cutting away as much paraffin or wax from your FFPE sample as you can. Then you expose the sample to chemicals to deparaffinize or remove as much paraffin from the sample as you can. After that, you start a lysis process where you decross-link, you heat the sample, and you try to free the nucleic acid for binding. Then you expose the sample to your column, where you try to bind as much nucleic acid as you can, as much DNA as you can to the column. Then you wash the column a couple times to remove any impurities or proteins, and then you add another buffer to elute that DNA off the column and collect it in an elution tube, where you then dry it down and it's ready for use. With the Ionic system, we try to simplify it. So we don't require, because we're using isotachophoresis, we don't require physical removal of paraffin. So you don't have to macro dissect or micro dissect. You can use a whole intact FFPE scroll to start. You add a lysis buffer, and then you really deparaffinize, decross-link, and lyse all in one step. You add the buffer to a tube containing your scroll and put it on a thermomixer. Then when you start an incubation cycle. Once the cycle is complete, you have lysate that you can then load onto an ionic chip on the ionic system. To demonstrate the advantage of the FFPE to pure DNA kit, we used a qPCR assay to measure yield and concentration. We used the QIAGEN multi-copy reference assay, or the MREF assay, and we compared extracts from the ionic system and a column-based kit. Across the x-axis are the results from 32 different FFPE blocks, where we took replicate samples and processed them through either kit. We took the samples that we used contained tissue from breast, colon, or lung. So we wanted to cover a diverse set of tissues to see if there were any tissue-specific differences. The y-axis shows the concentration achieved for either kit for each sample. And as you can see that for a majority of the blocks, the ionic system produces a higher concentration, typically around 3x, but as much as 10x in some cases. After demonstrating a superior yield from FFPE samples, we sequenced the samples to see if there are any differences between the extracts from the two kits. Since our focus is on the difference in sample prep, we decided to compare the extracts of either the column-based kit or an ionic system to the coverage profile obtained from high-quality reference DNA. The libraries prepared for sequencing were prepared using the Agilent SureMaster tumor hotspot panel. In the graph at the top, we plotted the log ratio of hits identified in the libraries of the same sample purified by each kit to the hits from the reference. The hits are grouped by chromosome across the x-axis. The ionic system is in blue, and the column kit is in yellow. The closer the hits, so the clustering of the data points is to the 0 line, the closer those hits are to matching the reference. You're really looking at the ratio of the extraction method to the reference sample. Across all chromosomes, you can see that the coverage of the hits for the samples purified using the ionic system are much more closely aligned around that 0 line, right? They're much more closely aligned to the reference sample. And to understand what was causing the underrepresentation of hits or targets in some of the chromosomes, we reanalyzed the data and looked for trends. And we noticed that some of the targets that were longer were unrepresented, as well as targets that had more GC content. And that indicated that for column kits, there was a bias towards fragment length or towards GC content that we didn't have with the ionic system. Our next step was to see how that variation in coverage, right, that kind of difference in coverage uniformity impacted the ability to detect variants. So we extracted DNA from 1, 2, or 4 FFPE sections per sample using the ionic system or a column-based kit. And then we performed the same coverage analysis as before, but we also looked at variant detection. For the ionic system, you can see that as you go down from 4 5-micron scrolls to 1 5-micron scroll, that you're still detecting at least 25 of the 26 variants. Really only in the 1 scroll sample did we see that 1 variant wasn't detected. However, when you look at the results of the column-based kit, you can see that even at 4 scrolls, you get variable performance. One of the 4 scroll samples, you could detect all the variants in, and in the other sample of 4 scrolls, you only detect 23 out of the 26 known variants. That also correlates with the coverage uniformity, right? You can see that those samples where we're not detecting variants, the coverage uniformity doesn't tightly match the reference. And so we think that with our technology, because there's no bias towards length or towards GC content, that you get a more reliable detection of variants, even as you go with smaller input, right, down to 1 5-micron scroll. Now I'll review the workflow and performance for our FFPE to pure RNA kit. Just as we saw with the DNA kit, the workflow for the FFPE to pure RNA kit is much simpler than that of the column-based kit. Just 13 steps in comparison to 33. You go through and actually count out all of the different touch points for each workflow. But what's most impressive is that because we're using ITP, we get both microRNA and mRNA from one process, right? So one run of the workflow gets you total RNA, whereas with column-based kit, typically you have to run through the workflow twice. So you'll take a sample, you'll split it, you'll run half of it through the 33 steps to get your mRNA, and then repeat that again to get your microRNA. So for total RNA, the Ionic system offers a simple and much faster path. As with the DNA kit, we compared the yield from the FFPE RNA kit to that of a column kit across 16 FFPE blocks. So we took adjacent replicates of each block, split it between the two methods, and then measured the yield using a qubit fluorometer and the double-stranded high sensitivity assay. You can see that for 15 of the 16 blocks, that the Ionic system outperformed the column kit. You can see that on the right, we've averaged the performance by tissue type. And again, you can see that the Ionic system outperforms the column-based kit in most cases. We also wanted to look at microRNA specifically. So we used an assay that targeted miR16 and miR21 targets. And we looked at the expression of those targets from that assay and converted that into an estimate of the concentration using a standard. And you can see that across the samples on the bottom, so there are 7 tissue samples. You can see that in most cases, the Ionic system outperforms a targeted microRNA column kit. Most noticeable to us is that for some of those samples, the material that was extracted using the column-based microRNA kit wasn't detected at all in the assay. Whereas with the Ionic system, almost in every case, there is material that's being detected. Next, we ran sequencing. So we ran next generation sequencing using the AmpliSeq Illumina Immune Response Panel to, again, look at any potential differences between the extracts of the column-based kit and our RNA kit. And so first, we looked at mRNA expression. And you can see that across the different tissue types, breast, colon, and lung, that the samples or the results of the Ionic system correlate well with the RNA column kit. So we're getting more yield, and the quality is comparable to the column-based kit. Or to say it differently, there's no negative impact on your downstream sequencing. The same is true with the microRNA. On the left, you can see that we compared the expression of target microRNAs between 2 replicates from the Ionic system, and the correlation is quite good, 0.98 Pearson correlation. And then on the right side, you see that the extract from the results from the Ionic system also correlate well with that of the targeted microRNA column kit. Again, we're getting more material, and there's no negative impact on our downstream performance. Now I want to introduce our newest product, the FFPE Complete Purification Kit. As I mentioned before, this kit can be used to extract DNA and RNA from the same FFPE sample using a simple workflow. Unlike many other all-in-one kits out there, our kit uses a simultaneous extraction, and that greatly simplifies the workflow. Traditionally, with other technologies, if you used a simultaneous extraction method, you often sacrifice yield because you effectively have to split the yield. But as I'll show you with our data, we get comparable yields, even though we are splitting the lysate and process. Here's a little diagram to explain this a little better. here you're looking at a portion of our fluidic chip, and you can see that sample 1 is being split, or I should say the lysate from sample 1 is being split into 2 wells of the chip. And in those wells will feed the sample into 2 microfluidic channels where DNA and RNA will be purified simultaneously. As we did with our previous kits, we ran standard QC assays to compare the performance of the complete kit to a column-based kit that was designed for DNA and RNA extraction. To do that, we took adjacent 10 micron sections from 16 FFPE blocks that contain tissue from brain, breast, lung, and colon. And we used each kit to extract RNA and DNA from those samples. On this slide, if you look at the left, you're seeing an average yield measurement from those samples as measured by a qubit assay. And on the right, you're seeing a measurement of beta-actin expression from the same set of samples using qPCR. And in both cases, we found that the results were similar between the kits. In some cases, the complete kit outperformed the column kit, and in other cases, the column kit outperformed the complete kit. However, if you look at the ratio of yields between the complete and column kit for each sample, and then take an average of those ratios across the sample set, you'll see that the complete kit offers a bit more RNA. So it has a slight yield advantage over the column-based kit. Looking at DNA from the same set of samples, the story is the same. The DNA yield from the FFPE complete purification kit is slightly higher for some samples and slightly lower for others. And looking at the overall performance across the sample set, the performance of the complete kit is approximately the same as that of the column kit. So they're basically equivalent. Since DNA is more stable though, we decided to take samples from a subset of the blocks and perform a second lysis that had an overnight incubation. And we just wanted to see if that was going to improve yield in any way. And the data here on the left shows that the average improvement across the sample set is better now for the complete kit. It's slightly better, but when you look at the right, you'll see yield is measured by qPCR. So you're really looking there at the amplifiable, or the yield of amplifiable material in the sample. And you can see that that's increased significantly for the complete kit. Across the sample set, there's almost 3 times as much material as what you're getting from the column kit. So with the FFPE complete kit, if turnaround for DNA extraction is critical, the standard workflow will give you a yield that's comparable to a standard column-based kit. But if you want to maximize your amplifiable yield from DNA, and you have the time, we include the option to run a second lysis with an overnight incubation. With that, I want to point out that the workflow of the FFPE complete kit is really the advantage here. It takes just 5 hours to purify RNA and DNA from a set of 8 FFPE samples, but only 90 minutes of that is hands-on time. In comparison to column-based kits, where most of that time is hands-on and the total time is longer, the complete kit cuts down the hands-on time by as much as 75%. To follow the new FFPE complete kit, we have several projects in development. In the near term, we are working towards the launch of new kits to support the extraction of DNA and RNA from larger amounts of cells, as well as from fresh frozen tissue. And then to follow that, we have several early stage projects that we are working to transition into full development projects for the future. If you have any interest in these near-term projects or longer-term projects, certainly reach out to us. We're always looking for new partners to collaborate with. And with that, I'll turn it over to our guest speaker, Dr. Shankar Mohan, who leads a team at Canopy Biosciences that has used all of our kits to purify DNA and RNA from their FFPE samples using the Ionic system. Thank you.
Hello, everyone. Thank you very much for your time today. My name is Sankar Mohan. I'm the Director of Operations at Canopy Biosciences. I'm really excited to share the experience of working with Purigen Biosystems Ionic kits today with you all. So, who is Canopy Biosciences? Canopy is a full service CRO. We have capabilities upstream and downstream of many workflows, and we are really well versed in working with client partners. We are professional, easily accessible to our customers from the receipt of samples through the service assays, data analysis, and delivery. Canopy is actually headquartered in St. Louis, Missouri. The Hayward, California facility, formerly known as Code Diagnostics, is a CLIA certified lab. And we have two sites in Germany that specializes in chip cytometry technology. In September 2020, Canopy was acquired and became part of Bruker family. So our overall portfolio is kind of listed here. This includes NanoString's Encounter and GeoMax services, immunohistochemistry and pathology services, as well as sequencing service lines such as RareSeq, microbiome diversity identity sequencing, whole exome sequencing, and RNA-seq. So we have evaluated the ionic systems for processing FFPE tissues by comparing the performance of each kit to our in-house traditional methods. So we kind of tested three kits here. The first one is Ionic FFPE to pure DNA kit, was compared against QIAMP DNA kit from QIAGEN. Ionic FFPE to pure RNA kit was compared to the FFPE RNA isolation kit from Roche. And finally, Ionic FFPE complete purification kit was compared against two kits, All-Prep DNA RNA FFPE kit from QIAGEN, MagMax FFPE DNA RNA Ultra kit from Thermo Fisher. So we did a head-to-head comparison of all these kits. For each kit, we compared the nucleic acid yields from several representative FFPE tissues from our inventory. And also we evaluated the workflow of each kit, including the total runtime as well as the hands-on time. So this slide represents the summary of FFPE to pure DNA kit versus QIAMP FFPE DNA tissue kit from QIAGEN. All of the extractions were done here at Canopy Biosciences in Hayward, California, CLIA lab. All these extractions were done by our experienced research associates. So we selected FFPE tissues of various size, tissue type, and age. We have used 110 micron section for each of these tissue. The graph here shows the yield per sample in a log scale. The light blue bar represents Ionic kit and the dark blue bar represents the QIAGEN kit. As you can see from the data here, all across all samples, but one sample, the Ionic kit really performed very well in terms of DNA yield. For one breast cancer tissue here, which was really small, the QIAGEN kit could not get any DNA, whereas the Ionic kit produced a significant amount of DNA. That is kind of very encouraging data. And also, if we look at it, overall, Ionic system delivered almost 3.7 times more DNA compared to the QIAGEN kit. This slide is similar to last slide, but here we compared the FFPE to pure RNA kit versus Roche FFPE kit. All extractions are done here at Canopy Biosciences, and we have used 110 micron section for each of these samples. Again, the graph shows the yield per sample in log scale. As you can see from the graph here, the Ionic system really outperformed the traditional method in 9 out of 13 samples. And also the interesting data is that the Ionic system produced a significant amount of RNA while the traditional Roche FFPE kit failed to get any RNA. This is important because sometimes the column-based methods fail to produce recoverable nucleic acid from smaller tissues. This is good to see that the Ionic system works really well when you use like smaller tissues like needle biopsies. So this slide is a comparison between FFPE complete purification kit versus MagMax versus AllPrep. So we kind of compared 3 kits heads on. All extractions were done here at Canopy Biosciences. Again, we used one 10 micron section for each of these samples. The top graph shows the DNA yield and the bottom is the RNA yield. Light blue bar is Ionic, dark blue is MagMax, and red is AllPrep kits. The top bar represents the DNA yield in log scale. As you can see from the data, for both RNA and DNA extractions, the results from Ionic kit was kind of comparable. So the other point here to keep in mind is that for Ionic kit, the lysates are split into half. Half of the lysate is used for DNA and the remaining half is used for RNA. Whereas the other 2 kits, you use the whole lysate for DNA isolation followed by RNA. The Ionic system really effectively provides comparable yield with the half the input amount of the lysate. That's kind of encouraging. So now we compared the workflow of each of these kits for a single run of 8 samples. So we listed here the time it took for each of the step. For lysis preparation, Ionic kit took 1.5 hours. MagMax was overnight, and AllPrep was only an hour. The RNA isolation time is more or less same for all of these kits. If we look, the DNA isolation, Ionic kit took only 1 hour 40 minutes. MagMax was 3 hours, and AllPrep took like 3.5 hours. But the interesting point here is the overall hands-on time for isolation of both DNA and RNA was only 1 hour and 30 minutes for Ionic kit, whereas MagMax and AllPrep, like it took 6 and 7 hours respectively. So it's interesting to note that Ionic kit takes only 1/4 of MagMax kit and even less compared to AllPrep kit. Now, what are the advantages and disadvantages of these 3 kits? So overall, the protocol is easy to follow for all of these kits. One of the advantage of Ionic kit is the DNA, the lysate for the DNA can be frozen and you can isolate at a later stage whenever we want. Whereas in AllPrep and MagMax, you need to do it same day so you don't have an option to store the lysate. With MagMax is a magnetic bead-based method, so they use a colored beads. The workflow is kind of a little bit easier because you can see the colored beads when you remove the supernatant. When it comes to disadvantage, the only disadvantage that I see with Ionic kit is you can only run 8 samples at a time, but you can do several runs on a given day. With MagMax and AllPrep, most of the time is hands-on, so it's very labor intense. For MagMax, you have to vertex the magnetic bead several times. That makes the beads kind of sticking to the Eppendorf tube lids, so you have to constantly kind of recover it. And also the bead drying time varied between 2 to 10 minutes. One has to constantly sit and watch to make sure that the beads does not get over-dried, because over-drying of beads, magnetic beads, leads to less yield in nucleic acid. For all prep, again, it's a hands-on, lot of labor-intense work, a lot of pipetting, a lot of centrifugation and everything. There is an incubation step of 3 hours for DNA isolation. One of the challenges we faced with all prep is that the deparaffinization of the tissue. So even after several incubation cycles, the tissue did not digest completely. This led to the spinning of the tissue becoming hard, and this led to kind of loss of tissue that kind of ultimately results in less nucleic acid yield. The other disadvantage of all prep kit is that the DNA has to be isolated on the same day. There is no option to save the pellet for DNA for future isolations. With the Ionic kit, it's automated. You can just load the lysates on the cartridge and you can walk away and do other work. In 90 minutes, you just go back and collect the nucleic acid. So it's kind of very simple. Now, coming back to the summary, we have seen the 3 kits has been evaluated here, FFPE to pure DNA kit and FFPE to pure RNA kit. As we have seen from the data, there is a significantly higher yield of nucleic acid by Ionic kit compared to the traditional methods. The workflow of Ionic kit is really simple and it's automated. You can just load the lysate and walk away. It's that simple. The DNA kit recovered DNA from a sample, whereas the QIAGEN kit did not. Similarly, the RNA kit recovered RNA from 5 samples, whereas the Roche kit failed. Coming back to the FFPE complete purification kit, as we have seen, it took only an hour and a half hands-on time, so it's really significantly lower compared to the other 2 kits. Also, even though we are splitting the sample, the lysate into 2 halves, but still the yield of DNA and RNA is higher compared to the other kits. That's all I have. Now I will hand it back to Elizabeth for Q&A section. And thank you very much.